Assalamualaikum and good day to everyone. My name is Muhammad Azzim Hakimi bin Khalid and my matric number is 2023-502255. Today, me and my team members are going to present about next-gen Halal Biotherapeutics, computational discovery of novel nanobody as IMP-13 inhibitor.
Antibiotic resistance has become a major global health problem. This is because bacteria are becoming more stronger and no longer respond to the antibiotic such as penicillin and also carbapenem, which is the last option that we use to treat the serious infection. This could happen because bacteria can produce an enzyme like beta-lactamase that will destroy the antibiotic and help them to survive. As a result, the infection are becoming more harder to treat and life is at greater risk. Carbapenem are considered the last resort antibiotic to use when all treatment are failed. However, these are now under treat because a group of beta-lactamase called imipinamase or IMP type of enzyme. These belong to the metallo-beta-lactamase MBL family, which requires zinc to function effectively. Among them, IMP-13 is particularly concerning because this enzyme is highly efficient at breaking down a wide range of beta-lactam antibiotics, including carbapenem. When bacteria carry a IMP-13 gene, they become extremely drug-resistant and make the infection become harder to treat. Even worse, this gene often carry on plasmid, which is a small DNA molecule that can transfer easily between different bacteria. This means the resistance doesn't just stay in one type of bacteria, but it can quickly spread in a hospital community and also across the border. Adding to the challenge, there is currently no halal certified treatment that directly target the IMP-13. This raises an ethical concern, especially for Muslim patients who may face limited treatment options due to the religious requirement. So, this is not only a scientific and medical issue, however, it is also a social and ethical concern. So, that's why we urgently need more research into halal biotherapy alternatives such as antibody enzyme inhibitor to fight this antibiotic resistance. Nanobody is a small and special type of antibody. It is called a single domain antibody. It can be found naturally in animals like camel and also llama. Unlike traditional antibody, nanobody are smaller, stable and easier to produce. Because of this advantage, nanobody are already being used in cancer therapy and also COVID-19 treatment. One of the exciting areas of this research is the uses of nanobody to help fight the antibiotic resistance. Nanobody can bind very specifically to the enzyme like beta-lactamase that bacteria use to destroy the antibiotic. So, by blocking this enzyme, nanobody can help to restore the effectiveness of antibiotic and make the treatment of antibiotic resistance possible again. Another important point is that nanobody can be produced using halal certified methods such as through microbiome system. These make them suitable for halal biotherapy treatment, which is especially important for Muslim patients who require halal compliant healthcare option. Nanobody are not only powerful in science but also offer a safe, effective and halal solution for the future treatment, especially in fight against resistant infection.
Assalamualaikum, I am Nurul Izzati binti Aziz. To investigate the interaction between nanobody and IMP-13, we implemented a systematic computational workflow. First, we retrieved the 3D structure of IMP-13 from the RCSB PDB. Then, we obtained a nanobody protein sequence via single-domain antibody database and predicted its structure using CollectFull. Next, structural quality and physicochemical properties were validated using mole property and expressy prot param. For interaction analysis, we conducted protein-protein docking using ClassPro, then visualized the dot complex in Chimera X. Lastly, we generated 2D interaction map using LeapPlot+. This workflow provides a robust in silico approach to support the design of halal-compliant nanobody-based biotherapeutics targeting antibiotic resistance.
This slide shows the result of our docking analysis. On the left, you can see the IMP-13 enzyme on its own with the active site open and ready to break down antibiotic. On the right, we can see our nanobody candidate attached near the active site. Even though it doesn't block the site directly, it binds close enough to possibly interfere with the enzyme normal function. This kind of binding can still stop the enzyme from working properly, either by changing its shape or getting in the way of antibiotic molecules trying to reach the site. Our analysis showed that the nanobody interact with key part of the enzyme like valine 25 through hydrophobic contact and histidine 197 through hydrogen bonding. These interactions help the nanobody stick tightly to the enzyme. Importantly, the nanobody was designed to be halal friendly. It's produced using microbial system, not animal, making it suitable for ethical and faith-compliant therapeutic use. This slide shows the result of our docking analysis. On the left, you can see the IMP-13 enzyme.
In focus on nanobody derived from camel as novel therapeutic to inhibit beta-lactamase, an enzyme responsible for antibiotic resistance. This project uses computational method to design nanobody, but the final goal is bioproduction that aligns with Islamic principles. 4 key halal considerations. First, source must be comes from halal and permitted under Islamic law. Second, all inputs in production must be halal compliant and free from haram substances. Third, process must follow halal pharmaceutical guidelines, ensuring the product is most suitable for Muslim consumers and certifiable. Lastly, production should reflect Tayyib values, meaning it must be clean, good, ethical, and lastly, environmentally responsible. This approach ensures not only scientific innovation but also religious and ethical acceptance in Muslim communities.
Today's consumers care not only about what works, but also what aligns with their ethic, faith and sustainability values. According to the State of the Global Islamic Economy report by Dina Standard and Thomson Reuters, the halal pharmaceutical market is projected to exceed $150 billion by 2030, driven by the needs of over 1.9 billion Muslims worldwide. Malaysia continuously to lead globally with halal certification standard like MS2424:2019. It's a Malaysian standard that outlines the general requirement for halal pharmaceutical as supported by JAKIM and Halal Development Corporation. At the same time, sustainable consumerism is pushing biotech toward ethical, animal-free and low-carbon manufacturing. Our nanobody-based inhibitor meet both demand. It's designed computationally, produced in microbial system without animal-derived component and avoid haram ingredient. It's aligned with global market growth and sustainability goals identified by the UN and documented by agencies like Salam Gateway and Frost & Sullivan. This is biotherapeutic reimagined, science that effective, ethical and halal. This research introduces a novel computational approach to designing halal-certified nanobody therapeutic, a breakthrough that not only contributes to antibiotic-resistant research but also pioneers ethical drug development. By aligning with halal principles, these therapies offer Muslim patients access to cutting-edge treatment without compromising religious value, promoting equity in healthcare access. Beyond individual benefit, this innovation fosters community trust, support global health inclusivity, and encourages sustainable consumer choices in biopharma. The use of nanobodies known for their stability, specificity, and low immunogenicity hold promise in developing affordable targeted therapies. Collaborations with halal certifying bodies and biotech startups can further accelerate the translation of this research into clinical and commercial impact.
The next step for this research is to move beyond computational analysis into wet lab validation, where the nanobody effectiveness can be tested in real biological systems. From there, we aim to continue through R&D and scale-up process to prepare the treatment for wider application. Future work may also involve clinical trial to ensure the safety and efficiency in human use. This will create a valuable opportunity for collaboration with halal certified bodies and pharmaceutical companies to develop halal biotherapeutics. In the long term, this project also opened the door for potential biotech startups focusing on nanobody-based solution for antibiotic resistance.
In short, we found a promising nanobody that can bind near the IMP-13 enzyme, which may help stop the antibiotic resistance. What makes it even more special that is, it's designed to be halal, no animal ingredient, fully ethical and made using microbe. This shows that we can combine science, sustainability and faith-based value in one solution. Hopefully, this can lead to a safer, more inclusive medicine for everyone in the future. Thank you for listening.